Hi, good evening. My name is Gabriela Sanchez. Today is Tuesday, December 8th, 2020. Today I'm going to be performing the experiment for lab number 8, and that's the carb fermentation testing. I have gathered all of my materials. I have the pheno red broths. I have my pipettes. I also have the Durham test tubes. My candle is already lit as well, and I have the glucose and all of the other powders on hand already. The first thing that I'm going to do is I'm going to get each of the vials. So they're already labeled, my pheno red broth base vials. They're already labeled. I have a total of 6. Number 1, S. cerevisiae. Number 1, S. epidermidis. Number 2, number 3, same for both. I'm going to get these ready first. I'm going to uncap the S. cerevisiae pheno red broth base vial. I'm going to pass it through the flame to sterilize it before I introduce anything to it. Once I've done that, this one is labeled S. cerevisiae glucose. So I have my glucose powder, and very carefully I'm going to pop it open and pour half of the glucose powder into the vial. Okay. All right. And I'll recap that. And I'm going to pass it through the flame again before I recap it. And I'm going to shake it to make sure that all of the glucose powder is dissolved. I'm going to do the same for number 1, S. epidermidis glucose. Uncap, pass it through the flame, and pour in the remainder of the glucose powder. Again, pass it through the flame before I recap it, and shake it. Make sure that all of the powder has been dissolved. I'm going to do the same thing for all 3 sets, with the difference of the different powders, the fructose, the mannitol. But for the sake of time, I'm going to proceed with S1, or number 1, S. cerevisiae and S. epidermidis. The next thing that I'm going to do is I'm going to uncap S1 cerevisiae, and I'm going to add the Durham test tubes into the pheno red vials. They've already soaked in alcohol. I've already let them dry thoroughly as well. So I'm going to uncap it, pass it through the flame to sterilize it once again. I'm going to grab one of the Durham test tubes that's already been out to dry, and I'm going to insert it with the opening facing down. All right. And that's in there. Before I recap, I'm going to place it through the flame once more. Recap, and I'm going to do the same for S1, or number 1, S. epidermidis glucose. And again, I'm going to do the same for all 3 sets, but just for the sake of showing what I'm doing. Pass it through the flame once more. Grab another one of the Durham test tubes that have been sitting to dry. Drop it in the vial. And we have to make sure that there's no air bubbles in there. And before I recap it, pass it through the flame once again. And then I'm going to recap it. So now that those, since we're doing those two right now, the Durham tube is already properly positioned in the broth vial. The next thing that we're going to do is, with the pipette that's been in alcohol as well, I'm going to transfer. So I have my both active cultures. One is the S. cerevisiae, the other one is the S. epidermidis. For the S. cerevisiae pheno broth, I'm going to uncap it, and I'm going to pass it through the flame. And I'm also going to uncap my live culture S. cerevisiae. I'm going to pass it through the lid through the flame. And I'm going to gather with my pipette. I'm going to gather, I'm going to grab 4 drops of the live culture. I'm going to shake the excess liquid and put 4 drops into the pheno red broth. Before I recap, like anything, I have to pass it through the flame. And do the same with the pheno red broth. Now this is ready to be incubated for 12 hours. I'm going to repeat the same process with the number 1 S. epidermidis glucose. Again, uncap and have my live S. epidermidis live culture. Pass it through the flame as well. And with my other pipette, I'm going to do the same. Make sure I have all the excess fluid out. I'm going to grab 4 drops and put it in the pheno. 2, 3, 4. All right. Before I recap, pass it through the flame once more. Once I'm done with the live cultures, once I've done all 3 sets, I'm going to refrigerate them for future labs. So I've put that through the flame before I recapped it. Now they're both ready to be incubated. So I'm going to repeat the process for all of 3 sets with the difference of the fructose and the mannitol powders. And I'll post a results video in about 12 hours or so. That concludes my video for today. I'm going to finish my experiment.